I'll give the floor to Rob from San Diego. Thank you for joining, Rob.
Thanks, Karina, and thanks to you, Gloria, and the other organizers for the invitation to do this. So, I was originally asked to describe microbiome data analysis in a 15-minute slot, and as you probably gathered from Gertus's talk just now, it's really a large and complex topic. So what I can do is give you a very high-level overview, given the time available. Microbiome science is a great example of what used to be a data-limited science, moving rapidly to an analysis-limited science. We're better and better at collecting all of the data to address these difficult topics, but analysis of all of that data is still a major challenge. To put this in perspective, back at the end of 2007, published in 2008, my lab in a single analysis doubled the number of microbial sequences in GenBank of a particular gene, the 16S ribosomal RNA, that's widely used as a taxonomic marker. And in this one experiment, we were able to simultaneously gain insight into microbiomes associated with lungs of cystic fibrosis people, with rivers across North America, of air sampling, and even of the Guerrero Negro microbial mat, still the most complex microbiome that's known on Earth. And what we were able to do was to take hundreds of samples from all these different environments and analyze them in one sequencing run, using a then new algorithm called Unifrac to relate all of those samples to one another, giving very nice clustering by sample type, and allowing us to analyze all these different all of these different environments simultaneously. So, in the study, we collected 500,000 16S ribosomal RNA sequences. At the time, the Sanger facility down the hall from my lab was still charging $8 a sequence. So if we had done it with traditional technology, the project would have cost about $4 million and taken about a decade. With the 454 technology, this was one of the first applications of for microbial sequencing. We were able to reduce that cost to $12,000 and to do the project overnight, as opposed to over the course of many years. And the key innovation was to use barcoded sequencing, where we introduce into each sample a DNA tag, in this case with formal error correcting codes, that allows us to tell which sample each of the sequences came from, without the physical separation that had been used prior to that. So this barcoding approach was really what enabled large-scale projects like the Human Microbiome Project, the Earth Microbiome Project that I lead with Jack Gilbert and Janet Jansen, the American Gut Project that we run out of UC San Diego, and its international expansion, the Microbiota initiative. But perhaps what's most remarkable is that to collect 500,000 sequences that would have cost $4 million in 2007, reduced to $12,000 in 2008. Today, that amount of sequencing costs you just 75 cents on the latest NovaSeq 6000 instrument. And a lot of what drives the study designs that we're excited about ourselves is this reduction in cost, because if it's going to cost you millions of dollars to analyze a sample, you're not going to analyze very many samples. But if it costs you less than $1, that dramatically expands the possibilities for characterization. The problem then, though, is that you have to analyze all of that sequence data, and this itself is a major challenge. So, for example, in the Human Microbiome Project, the large consortium that I was a part of, with something like 400 researchers across the US, collected 1,500 human genome equivalents worth of data, about 4.5 trillion bases. And so, so the great thing about this is we had an unprecedented amount of sequence data. That was also the terrible thing about it. So here I'm just showing you the first file of data from the HMP. This is the first 0.1% of that file, and this another 17,000 just like it. And it's very difficult just looking at the sequences, figuring out what's going on. this happens to be an oral sample, but figuring out what sequence signatures related to the mouth is very challenging, and let alone anything about the phenotype of the subject. And this kind of thing is increasingly a problem, not just from a research project, not just from a research perspective, but also from a clinical perspective, because now there's increasing opportunities for individuals to get their microbiome sequenced. And it's certainly a physician's nightmare that you're going to show up in their office with a huge smile on your face and say, hey, doc, I have this great news for you, which is that I had my microbiome sequenced. Now I have this list of 1,000 species they found in my gut. With all that data, what are you going to do for me? And I mean, what's your physician going to do? Refer you to colleagues in psychiatry for being crazy enough to think that they could do something with all that information in the very brief time that you have together. So a lot of what we do in my lab is try to make it not crazy that you could do that sort of interpretation, even in a very brief time, essentially by automating this workflow of amplifying many samples, characterizing them either by 6-genus ribosomal RNA profiling to look at marker genes, or by shotgun metagenomics, taking the sequences, align them, building phylogenetic trees, and then clustering the communities relative to one another. And so as time has gone by, we've replaced older technologies with newer ones. And all of this is encapsulated in a pipeline called CHIME, Quantitative Insights into Microbial Ecology, which is free, it's open source, it runs anywhere from your laptop to the Amazon cloud, to some of the largest supercomputers on the planet. And in general, UC San Diego and the Center for Microbiome Innovation that I direct puts a lot of effort into driving forward algorithms and analysis. Everything from workflow engines like Cheetah and GNPS, which is its equivalent for metabolomics, where both of those are backed by giant databases spanning hundreds of thousands of samples. Visualization tools like Emperor, analysis tools like GenePattern, Cytoscape, and so forth. So anyway, applying all of these tools is a bit like flying a plane, right? I can't tell you how to apply all these tools safely in the course of 15 minutes, but I can basically give you the safety briefing and give you a sense of what some of the pitfalls are and what you need to watch out for in designing your plan to do these analyses. So starting with safety briefing, one really important component is it's critical to listen to crew member instructions, and all of these microbiome tools are designed for specific purposes. It's really important to read the documentation and papers written by the authors of each tool, and then listen to colleagues who use them frequently. Over and over again, we see tools being applied to data sets that they don't apply to, or answering questions that require a different statistical model, or that kind of thing. And it's definitely worthwhile just getting a sense of what you will and won't be able to do there. So for chime2, for example, this very extensive user documentation that you have access to. There's also a lot of papers that we and others have written that basically cover what the strengths and limitations of different approaches are for microbiome data. The second point is that you want to have a clear exit plan in mind in case of emergency. So, for example, sometimes you're going to see a very clear and beautiful visualization like this. For example, integrating a Clostridium difficile infection patients with the Human Microbiome Project, where you have very clear clustering. But a lot of the time, you'll just get some mess where you have no clearly separated clusters, and it's very difficult to understand what's going on. So especially if you have a clear hypothesis up front, and being able to structure the analysis to test it will make your job a lot easier. And in this case, the animation didn't play very well, but basically the idea is that let me see if I can play that again. basically the idea is that if you have something clear cut like this case with C. diff, where you start off with people with a very dysfunctional microbiome, and you want to see whether on fecal transplant their microbiome becomes functional, so you'd expect it to move down to the stool distribution in brown at the bottom there. If I play that again, you can see that the traces for all the people who got fecal transplant move down into that healthy region. And so if you have a very clear-cut question like this that you're framing up front, it's a lot easier to see whether you got the result that you were that you were expecting or not. Third point I want to make is that being prepared upfront typically leads to better outcomes. And so for example, having all of your metadata in place, having a good sense of how things are going to be tracked, and using tools that allow detailed provenance tracking of every step essentially gives you a black box recorder that allows you to understand what happened in your data set and where each particular result came from. And for example, chime2 and Cheetah and Jupyter Notebook and other tools automatically track provenance. They let you reproduce the complete workflow, and they let you understand what happened if your analysis went wrong, as well as being able to replay it with new data sets that you want to incorporate in there. Related to that, one thing that's really important is don't ignore issues with your data. So if there's a problem with your data, don't bury your head in the sand, because the analysis probably isn't going to work very well. And a nice example of this is a recent paper that we did linking the microbiome to cancer, led by Greg Poor, a very talented MD PhD student in my group, where on the left-hand side, what we're looking at is a preliminary analysis where samples collected by different sequencing centers look completely different from one another. Whereas on the right, what you can see is that after detrending, those are all brought back into the same region of principal coordinate space. And so being able to precondition the data made the difference between, so being able to precondition the data made the difference between not being able to see and being able to see very good separation according to clinical phenotype. And so getting those kinds of things straightened out up front can be really important for the analysis going well. Let's see. Another really important point is that if your data are effectively on fire, like if there are massive problems with it, don't continue with the analysis, because you can waste an enormous amount of time dealing with a data set that's fundamentally not analyzable. And so, for example, if you do your analysis and you get something like this, where there's no separation at all between the red and the blue points, probably what that's telling you is that there's no signal and you should not proceed. You should work with some other data set instead. And in fact, many microbiome data sets contain no pattern at all. You could spend years trying to see a signal that's not there. And we see this a lot with graduate students who basically have one data set to analyze, and they're going to keep at it no matter how little there is to see in that data set. So with that, I'm going to wish you a good, safe microbiome voyage. I've gone over some of the considerations for what you want to think about in these analyses, and I hope you have a good time doing it. So with that, I'd like to briefly thank the many amazing members of my lab here at UC San Diego, our various sources of funding, including the tens of thousands of members of the public who've contributed to our crowdsourced efforts. And finally, thanks for inviting me to do this. I'd be delighted to answer any questions you have. Thank you.
Thank you very much, Rob. Questions, anyone? Rob, you mentioned how, oh, I see Alex has your hand raised. Do you want to hop in, Alex?
Yeah, Rob, thank you very much for a nice talk, very interesting and very stimulating. For the young people and young scientists in Africa, I mean, when it comes to microbiome data analysis, is sometimes a little bit of a problematic because it requires a lot of time and then attention, of course, and reflects exactly what you said. knowing exactly what you want to do and then trying to get it. But sometimes you get into a lot of hurdles and you have no idea what to do. What are some of the strategies that young scientists and the students and then researchers can take advantage of to really to be able to build on their competences when it comes to such an area? Because you can have huge data, but if you can't analyze it, it's as useless as anything. So maybe you want to give it a touch on something like that, Rob.
Yeah, absolutely. So one of the nice things about the way the microbiome tool ecosystem is developing is that there's a lot of forums where you can ask questions interactively, where, for example, for both Chime and Cheetah, there are very active communities where typically you'll get a response to your question within 24 hours. It's always, the best advice I can give you is don't be afraid to ask. The worst that can happen is that it'll take a while to get your question answered, but frequently asking a question on one of those forums can save you months of trying to explore it on your own. The other thing I'll reiterate is that the documentation and also papers that are written about how to frame the analyses and what tools to do and so on, what tools to use for particular tasks are also often very useful and very frequently published in open access venues. So, for example, M Systems has published a lot of those types of pieces, and that also helps by avoiding avoiding charges for being able to download and read the paper. So there's a lot of free resources out there, but the best is always talking to people who are already engaged in it. So I think networks like this network are going to be especially useful for fostering that dialogue and building up a community of practice.
Yeah, so there's a bit of a related question in the chat, which is asking, will the Chime tell you what data set you should use and how? I guess it will help you to choose, right? But do you want to comment on this?
Yeah, none of the tools that are available really tell you what samples should you use for your analysis. That's still very much a judgment call, because depending on your biological situation and depending on what range of biospecimens you're looking at, like whether it's only human or whether it's also environmental biospecimens of different types, what you expect to see in terms of variation in the data is going to be very different. And especially if you're doing something really new, the whole point is that no one knows what the answer is. So you're not necessarily you're not necessarily going to have a good idea up front. Exploratory analysis, which is things like principal coordinates analysis and other views of the entire data set, can be very helpful for figuring out if there's some structure in your data that you didn't expect. So, for example, if you're expecting all your data to look more or less homogeneous, and then you see two clear clusters that are unexplained, that's when you want to start digging into it and ask, is there a problem based on the preparation of the samples or based on the storage conditions or that kind of thing? And very, very frequently, when we see unexpected clustering in the data, we can track it back to some sort of problem with the experimental procedure. But it's very much a judgment call at this point, and a lot of those considerations are covered in some of the papers that I mentioned, like the Microbiome 101 for clinicians, for example, goes into that sort of thing.
There is a question related with the update of the reference databases for species level identification for 16S applicon, and if you think this is a problem, the low rate of update.
Yeah, it's a problem. Remember that the amount of funding that we have to update those databases is $0. So it's very much a spare time project that gets done as available. It would be really good if one or more of the funding agencies that have rejected proposals to do that on a more regular basis would actually fund them. But as it is, it just takes a long time where there's no resources specifically devoted to it.
So there's maybe as a last question, there's about opportunities of training to use these tools. I think as you already said, this is chime is freely available, right? Are there training sessions that people can enroll?
Yeah, there are virtual workshops, and you know, back before the pandemic, when travel was possible, we used to very frequently do workshops in different places around the world, including in Africa. So we are hoping that the pandemic situation resolves before too long, so that those in-person workshops, which tend to be a lot more productive, are possible again. Although given the current worldwide surge in COVID, that's probably going to be a while off.
But there are currently no like training sessions on like recorded or something that you did.
Yeah, there's extensive recorded materials, and there are also online workshops. So if you go to the if you go to the Chime2 website, there's a link that basically takes you to workshops where you can register and do online training, as well as pre-recorded tutorials for a lot of the stuff.
Okay, thank you very much, Rob. We'll have to make a break. Emma, do you want to say something?
No, we have 10 minutes to go on break. Yeah.
Yes. So let's be back at 35 minutes. So in 10 minutes, I'll write specifically the time in the chat.
Noted. Thank you.
Thank you, Rob. I know it's very, very early here, and to everyone, thank you very much for today. And I know it's been challenging for many people all over the world, and so thank you, Rob.
Sure. Thanks, Karina.